Our next speaker is Katarina Vucicevic, Professor of Pharmacokinetics, University of Belgrade, Faculty of Pharmacy. She has extensive experience in the academic environment and clinical research projects focused on pharmacocentric modeling and simulations. Understanding how drugs interact with biological systems is paramount for informed and effective therapeutic strategies. The focus of the presentation is the integration of data-driven approaches in deciphering the complexities of drug behavior within the human body. The talk will highlight the power of advanced statistical methods and data visualization in extracting meaningful insights from pharmacokinetic and pharmacodynamic data. Real-world data and developed models will be showcased to exemplify the application of the methodology. Katarina, the stage is yours.
Okay, thank you very much for a very kind introduction. At the beginning, I would like to thank DigiHealth and Damian for a kind invitation to be one of the speakers at this conference. And as you heard, I'm coming from academia, so my speech will be kind of different from what you are used to here at this conference. So at the beginning, let me just show with you the university and the faculty pictures from where I come from. So I'm affiliated with the University of Belgrade. It's the oldest university in Serbia, and it has been founded back in 1808. And under the umbrella of the University of Faculty, there are many science institutes, university library, 8 centers, and more than 30 faculties. And one of the faculties is the Faculty of Pharmacy. And we are the oldest faculty of pharmacy in Serbia, and not only that we are a higher educational institution, but we also do various healthcare services. We are involved in publishing, professional activities, and of course we do a lot of research, and our research is multidisciplinary. And now let me introduce myself from another perspective. I'm a pharmacist by training, but I do data modeling. So I'm sure it sounds very strange to all of you, but we are trying our best. And I have to tell you that in this field of pharmacometrics, at the departments of pharmacometrics in the pharmaceutical industry, there are a lot of pharmacists. And maybe it is due to the fact that the pioneers of this field were affiliated with the Faculty of Pharmacy. And we do a lot of studying about the drug, and we know how the drug behaves in the body. So this is probably one of the reasons why there are many pharmacists. Nevertheless, there are many engineers, mechanics, statistics people. So you know, each of us, we bring something to the table, and at the end we have a pretty good meal to serve. So this is the story about myself and about the university. So I'm going to tell you a little bit about the drug and how and what is the journey actually of the drug in the body, and how some data are being generated through this interaction, how we analyze this data, what are the models that we create, and how these models can be used to inform dosing strategies and therapeutic strategies. So we can say that a disease or a medical condition is actually a condition where there is a deviation from something what is considered normal or physiological, either structure or a function. And we basically give medicine in a specific dose in order to fix these deviations, in order to prevent some interruptions, perhaps. So we give a drug in a specific dose, and we expect some effect. And this is what is called pharmacodynamics. So basically, we have an effect which can be quantified. We can measure it. We can say, okay, what is the blood pressure? What is the level of the glucose? What is the pain that we feel at some scale of the pain? So we can measure, we can quantify these effects. Similarly, we can also quantify adverse effects, which can appear as well. On the other hand, what body does to the drug is actually something that can be quantified. And this part of the story is called pharmacokinetics. So we can basically measure how the drug is moving through the body. And this movement of the drug throughout the body, we do through measuring the concentration of the drug in the blood. So the concentration is changing over the time, and this tells us that something is happening with a drug in the body. So we have this bidirectional interaction between the body and the drug. So if we want to study this interaction, we have to use some models. We have to find a way how to present the body and how to present the drug. So let me show you what is the model of the body. So this is the body. For us, this is the body, very simplified version of the body. And this is the drug. The drug enters the body, and then the drug leaves the body. So this is our very simple version, very simple model of the body-drug interaction. On the other hand, we can use a bit more complex version, where we can basically divide the PK processes and the PD processes into various compartments. And let me tell you, this is still a very simplified version of the body. So the body is very, very complex, and no matter which model we use, we are still doing some kind of simplification. So the whole point is that we give a drug, the drug moves through the body, we measure the concentration of the drug in the blood. The whole purpose is for the drug to get to the target, to the receptors, to have some interaction. Then we have the change of some biomarker or some response that we can measure. And why it is changing? Because the pathology actually did some deviations, and then we have effect. We have this clinical effect, beneficial effect that we want to obtain. Okay, so the clinical trials are a gold standard for drug approval, and they do a lot of research about the PK and the PD of the drug. And the whole point of this process is, at the end of the road, we get the approval for the drug to be marketed, to get on in everyday practice. And then the drug is being registered for specific indications in the specific dosing regimen based on the evidences. And the evidences are nothing more than the results of the data analysis, which is being generated and conducted from the clinical trials. So what are the data that are being generated? Well, there are a lot of information about the dose, about the dosing regimen, about the characteristics of the subjects in the studies, whether we talk about patients, about volunteers. We take some information concerning the concentration of the drug in the specific time points. Then we also take the levels of the biomarkers in the specific time points. So we have this longitudinal data. And then we also have the effects that also can change over the time. So we have a lot of data that is being generated during the clinical trial. And this is the data. So basically, it's a bunch of numbers. Okay, here they're structured in some form, but we have a bunch of numbers that we want to analyze in a way to form clinically meaningful relationships and to extract some useful knowledge. And this useful knowledge is what are the evidences for the dosing regimen of the particular drug. So how we do it? So we have some data, and when we talk about PK or the PD of the drug, we want to fit some models. So we have here some basic mathematical equations, some differential equations, and we're trying to fit these models to the data that we observed. And while we are doing it, we are actually estimating some parameters. And these parameters can explain the PK or the PD behavior of the drug. We sometimes, and it's very important to go actually a step further and to try to connect these two parts. So that's why we sometimes develop exposure-response models, or we create PK-PD models. And based on these models, we have evidences and we have the information about how to dose a specific drugs. Okay, this is like a small introduction. Then let's talk about variability. The previous speakers mentioned something about variability. So if you just look around the people in this room, we are one sample of the people who attend this conference, and we are pretty much different. We are all unique individuals. And this individuality can represent a challenge or can be an issue sometimes. And usually it is, because we have to address this variability. So as I said, we have a sample, right? We never study the whole population. So we have a sample during the clinical trials, and we study this sample, and we hope that it will provide the information about the population. So for example, if we study this group of people here, we wonder, can we be a good representative of all attendees of this conference? And we can, judge, different, ways how to get to the conclusions. So this is the same thing that we do during the clinical trials. So we study the drug, and basically the criteria for the drug development phases are very strict. So we have a very strict criteria which participants can be involved in these studies. And based on it, we license the drug. However, the situation in the practice is pretty much different. So the patients who are taking the drug can be quite different. They're polychromatic, and they're not monochromatic as they were involved in the clinical trials. So we usually need more information how a specific drug performs, behaves in some subgroups or at the individual level. So the question is, how different or how similar are we, and based on what? So in this context, we talk about differences in the pharmacokinetics, in the concentration, and in the effects which are being created when one dosing regimen is being given to different people. So the magnitude of this variability is very important, and we have to address it. We have to quantify it. We have to, see what are the reasons why people behave differently when the same dose of the drug is being administered. So the challenge is because our patient or some subgroup of patients may need different treatment approach, or they may require a different dosing regimen compared to the usual, to the typical patients, to average patient, which is being involved in some clinical trial. So one size fits all usually doesn't fit anyone. So this is the situation that we have time to time. So what are the ways to quantify this variability? Traditionally, it's being done using very a simplified approach. So we actually study the drug in some group of patients. So we get a lot of data per each of these patients. We fit the model per individual data. We get individual parameters. And then we summarize these values, we find some average or median value, and we say, okay, this average or median value is something which applies to the whole group of patients who were being studied in our clinical trial. So from individual level, we go to something which is called population or group level. But this is not good enough because the data that we collect are they have some issues, they have particular characteristics, so we cannot always have this approach. For example, if we measure the concentration of the drug, and we have only one level per individual, so our ID number 5 in this study gave us, we obtained the blood level, which is this dot. The question is how we can fit a model through this dot. And how many models we can fit? Too many. So this is not, the way how we cannot analyze this information using the traditional approach. And what if our patients are, they have too many variables, and they're different by different levels. So we cannot form uniform groups of patients. What if some data are missing? Should we exclude this individual or not? What if the level of the concentration or some biochemical parameter which is being measured is below the limit of quantification, or the assay was not able to tell us what is the concentration level, but we know, it's below the limit of the quantification or above the limit, and so on and so on. So we have many issues which limit this traditional approach. So that's why we use something which is called population modeling or nonlinear mixed effects modeling. So in this approach, we can actually simultaneously fit all data together, so fitting data from one subject in the perspective of all the others. So our previous individual who gave us only one blood level, we can see this level in the perspective of all others. So we can when we combine all these levels together, we understand something about the behavior of this drug. And another thing is that we can study very heterogeneous population, and we can see what are the resources, what are the sources of the variability that we observe at the drug level or in the effect. So from the population level, we are going to the individual level of the subjects. So what I mentioned already is the variability which is called inter-individual variability. So why persons differ, why we are different? Well, we can be different due to many reasons: age, body composition, gender, and so on and so on. And this is one level of the variability. But we have to think about other levels of variability which also exist when we obtain clinical data. Sometimes we can see that the behavior of the drug changes over the time. And it is called inter-occasional variability, and it is very common in oncology, for example, when the drugs are given over certain cycles. So we see that the behavior of the drug or the response is changing over the time. So sometimes it's also very, very important to address this kind of variability. And there is another story which is called residual variability. So it is how much our measured level is different from the concentration or some response which is predicted by our model. So why we have deviations from what we measure to what our model tells us we should obtain. So at least we have these 3 levels of variability, which would be very, very good that we can separate and that we can quantify. And actually, using this approach of nonlinear mixed effects modeling, or so-called population modeling, or which is abbreviated with non-MEM, which is also the name of the software, gives us opportunity to address variability, to address each of these levels of the variability, to fit the model to the population level, also to get individual parameters of the model. And this approach is a golden standard for studying variability, and it's being recognized by the regulatory agency, by the FDA, American, and also European medical agency. And that's why in almost each pharmaceutical company which is doing drug development, they have a specific department where they do modeling, and they use primarily this kind of tool to analyze the data and to extract some information. So this population modeling is actually a pharmacostatistical modeling approach. And we have so-called structural model, and this is the model that I showed you before at the beginning of the presentation. Then we have something which is called regression model, and it's the model which tells us and which explains the correlation between various characteristics of the patients, of the therapy, and the parameters that are being estimated. So we explain the variability using these equations. And then we have the statistical part of the model, which tells us a lot about so-called random effects or inter and residual or interoccasional variability. So more or less, it looks something like this. So we have a population model, population level of the parameters, then we have individual level of parameters, and they have certain variance, and then we have also the variability in the concentration, so it tells us why the level that we measured is not exactly as our model predicts. So the software we are using actually employs these nonlinear regression techniques, and it optimizes fixed and random effects or parameters, and it uses maximum likelihood approach. There are different estimation algorithms that you can adapt, that you can use when you perform the estimation of the parameters. And basically, this is just some structure and some equations that tells us about the population method. And this is the likelihood function that we want that we are trying, that the algorithm actually tries to find the value of the parameters where this likelihood function will have the maximum value. And at the end, we estimate the parameters, and we have a certain uncertainty about these parameters. And of course, we have to see how well our model fits our data, so we use various diagnostics numerical or graphical diagnostics to evaluate how good our model is and are we satisfied with what we obtained at the end of the story. So basically, the whole field about this modeling is called pharmacometrics. So we are measuring the pharmacology of the drug, and we're trying to develop some models that will explain, that will be used to predict, that can be used for some simulations, and so on. And this approach is implemented basically in all phases of the clinical drug development, and as I said, it's being recognized internationally. So models can be descriptive, so they can describe something, they can describe the behavior of the drug in a simple way, simple way with certain equations, with some parameters. It can be explanatory, so they can explain something which is unclear in the data that we obtain, and it can be predictive. So we can use these models to predict the time course of the data behavior under some arbitrary, for example, dosing regimen or some hypothetical clinical scenario. And then we can say, okay, this is, we should change the dosing regimen when we administer this drug to the patients who have co-therapy and who are overweight or underweight and so on and so on. So for the end, I would like to share with you several examples of these models. So this is one very, very simple model and very simplified approach. But the story is that patients who have epilepsy, they take anti-epileptic drugs. And we know that epilepsy is an episodic disease, which means that we have phases with attacks and no attacks. And when the patient is taking the drug, we are not sure, are we, targeting the therapeutic concentration of the drug, or simply the patient is in the phase where no attacks. Very often, in order to control epilepsy, patients are required to take additional treatments. So they have 2 or sometimes 3 anti-epileptic drugs at the same time. So that's why patients who are on therapy of these drugs, they are required time to time to go to their medical doctor, and doctor requests blood sample, and he wants to see what is the concentration level of the particular drug. So you can think that when person goes to the doctor and gives blood, we actually obtain only one concentration, only one level. And we try to use these data and to analyze them in order to understand why drug levels are different from patient to patient, why we have such a huge variability in the data, and we try to explain it in the quantitative way. So we use data from approximately 130 patients, and 1 to 2 blood samples or concentrations of this particular drug were available. And after fitting the model to this data, what we actually understand at the end is that we have several very important factors who contribute to the variability in the parameter, and the value of the parameter actually drives the concentration. So not only that we understood that the body weight and the co-treatment with another drug and the total daily dose of this drug that we were studying is important, but we also found the equation how these factors influence the specific parameter. So each of the parameters which are estimated during this modeling is estimated, as I said, with a certain uncertainty, and that's why we can calculate 95% confidence interval. But also, we evaluated the variability. So we have this variability at the level of the drug, of the assay, and also at the level of the different persons. So this is the coding we do. So we code the model, we do some coding, and we're giving the instructions to the model how to use the data and what to do, what to perform, and then when we have outputs, we evaluate how good model it is, and then also using some graphs, we see how well fit we have with what we have observed. So at the end, everything which is written here is actually our model. So these equations here, and these parameters here, and this equation here, everything is called, this is our model. So when we know this behavior of the drug, we can use it. We can use it to adapt, to adjust dosing regimen. So at the beginning, when you have a patient, we can say, okay, what is your body weight? Are you taking, some additional drug? What is the daily dose? And based on it, we can say, okay, for you, probably good dose would be this and this. And then also, at a certain point of time, when the drug concentration is available, we can use this model to re-estimate individual parameters and to say, okay, if we want to obtain a certain therapeutic level, we should change your dose to this amount. So this is the application of the models. So we understand something of the variability, but also we can use it in a way to adjust dosing regimen. Okay, I had another example, but due to the time constraints, I will just skip it. And I will show you another very short example. This was one very, very interesting study. It's being performed on pregnant women who are living in the areas of Africa. And as you understand, there is a high risk of malaria. And not only the risk is for the woman itself, but also for the fetus and for the newborn. And we know that pregnancy by itself is a condition which changes some physiological parameters, and it can influence the behavior of certain drugs. On another hand, in the areas of Africa, we know also that there is a high proportion of HIV-positive persons. So we selected a sample of women who were pregnant, but who had at the same time HIV. So they were taking some additional drugs in order to treat HIV. And these drugs are known that they can influence the capacity, the function of our metabolic enzymes. So it can change the behavior of some other drugs. So we have some interaction between drugs. So in this particular group of patients, we have a hypothesis that the pregnancy by itself and the co-treatment with the HIV drugs probably can influence the drug for malaria, which can be used as a prevention because we have a high risk of malaria. And this is what we actually confirmed based on the study. And we saw that less than 4% of women who are taking antimalaric drug to prevent malaria and who are HIV positive do not achieve the level of the antimalaric drug which is needed to protect them from malaria. So basically, they're taking the drug, and this drug is not doing anything. So we said, okay, can we do something more? And we developed a model. We developed one PK model, and we used this model to simulate different dosing scenarios. And as a previous speaker said, we have to find a way how to communicate our data. So this is the way how we were communicated our results with the doctors. So we said, okay, the dosing regimen, which is the traditional regimen, will not work for pregnant women who are HIV positive. So we are giving you different scenarios, and we are simulating the concentration time profile based on the different dosing regimens. And in order to get the preventive drug level, you have to think about giving drug in either this or this or this or this dosing regimen, because the first four will not give us the results that we want. And based on these results, I hope that already some new clinical trials started that will confirm what we have obtained during our study. So take-home message from all this story is that at the moment we have evidence-based medicine and something which is called population-based dosing regimen. In the past, we were treating patients based on the symptoms and based on some empiric, on some experiences that we have. But we hope that very soon we will have precision medicine and that we will use models to inform dosing regimen. So at the end, what is a medicine? I mean, are we talking about science or are we talking about an art? And I like very much this citation, this quote, which says that medicine is a science of uncertainty. And indeed, when we estimate the parameters, we estimate it with a certain uncertainty, but also it's an art of probability. So we use various, probability distributions when we talk about the parameters. So you can choose. Does one size fits all? You can choose among different, sizes, or you will go to your tailor and ask him to take your measurements in order to create the suit which will fit your needs. That's it. Thank you very much. I think I was a perfect fit, 30 minutes. So if you have some questions, or maybe we can leave it for later. Yes, thank you very much.
Thank you very much, Katarina, for sharing your experience with us. And can you please take your certificate? It's right here. Thank you.